One.
Good evening. I'm Colin Lynch with Sunnybrook's Board of Directors. It is my pleasure to welcome you to our speaker series discussion on clinical trials. Before we begin, I would like to take this opportunity to say that at Sunnybrook Health Sciences Center, our mission is to care for patients and their families when it matters most. Sunnybrook also cares about our collective responsibility to achieve reconciliation with Indigenous peoples who have lived on this land for generations upon generations. We acknowledge our campuses are located on the treated territory of the Mississaugas of the Credit, First Nation, and ancestral and traditional lands of the Anishinaabe, Haudenosaunee, Ojibwa, Chippewa, and Huron-Wendat nations. Today, this land is home to many First Nations, Inuit, Métis, and urban Indigenous peoples from across Turtle Island. We value their contributions past and present, and we are grateful to work in the community on this territory. So, why are we discussing clinical trials this evening? Well, Sunnybrook has a large and diverse research scope that includes clinical trials, which are important to all research and clinical programs at Sunnybrook. Clinical trials help advance medicine and ensure our patients have access to some of the most innovative therapies and procedures. The Sunnybrook Research Institute plays an important role leading this work, and you'll be hearing more about that from our speakers this evening. Tonight's discussion will try to answer some key questions, especially for people who might be thinking of joining a clinical trial. How do clinical trials work? And why do they matter? We have an incredible panel of experts lined up for you to address these questions, and we are very fortunate to have Lisa Di Prospero moderating this event. She is Vice Chair of Sunnybrook's Research Ethics Board and Director of Practice-Based Research and Innovation. In addition, Lisa is also Director of the Sunnybrook Hub for Applied Research and Education. This is just a snapshot of her biography. As you can see, we are in very good hands having her moderate tonight's panel discussion. Lisa, thank you so much for being here. I'll now pass the microphone and event over to you.
Great. Thank you so much, Colin, and good evening to all of you who are joining us remotely. Tonight, our talk is called Thinking of Joining a Clinical Trial, How They Work and Why They Matter. As you'll learn shortly, clinical trials are research studies that test a medical, surgical, or behavioral intervention in people. Interventions include, but are not limited to, drugs, vaccines, devices, or procedures. At Sunnybrook, we have more than 500 active clinical trials that patients and participants can become involved with. These trials help us offer the most innovative care to your patients and to our patients, and are integral to our vision to invent the future of healthcare. Tonight, we are so lucky to have a great lineup of speakers for you. Dr. Neer Lipsman will kick things off by describing how clinical trials help to advance medicine. Mayalith Linus will then answer some common questions from clinical trial participants. And then Dr. Brian Murray will discuss how ethics help guide clinical trials. We'll set aside some time at the end of the evening for you to ask our panel of speakers some questions. Thank you to everyone who has already submitted a question online, and please feel free to also send your questions while the presentations are happening through the webpage. We may not be able to get to every question, but we will do our very best in the time that we have available. So to begin the lectures, I'd like to introduce our first speaker, Dr. Neer Lipsman. Dr. Lipsman is a senior scientist with the Sunnybrook Research Institute and a neurosurgeon. He is also director of the Harkale Center for Neuromodulation and co-director of the Focused Ultrasound Center of Excellence at Sunnybrook. Dr. Lipsman is an associate professor in the Department of Surgery at the University of Toronto. An early adopter of focused ultrasound, he is looking to bring non-invasive solutions to patients as quickly and safely as possible. He leads the way, collaborating with experts in a wide range of disciplines, from neurodegenerative disease to psychiatric disorders and oncology. This approach has led him and his team to several world first class clinical trials using focused ultrasound, including the first experience of blood-brain opening in Alzheimer's disease, ALS, and primary and secondary brain tumors. As you can see, we are very fortunate to have him here this evening to talk about the important topic of clinical trials. Dr. Lipsman, thank you for joining the panel, and I'll send it over to you.
Thank you so much, Lisa, for the kind introduction. I'll just start sharing my screen, but before I do, I just want to thank Sam and everybody and Idella and everybody for organizing this evening. Thank you so much, Colin, for joining us as well. It's an absolute privilege to join, and particularly to join alongside Mihalis and Brian, with whom we work very closely on a lot of the work that you'll see that I'll discuss in the next 20 or so minutes. So we do exist, as you'll see, in a very unique ecosystem here at Sunnybrook, where not only we have the great privilege to look after patients, but to really advance the future of brain therapeutics and develop new ways of treating the brain. So without further ado, let me share my screen. And I'll just ask the requisite, can you see my slides question here? Yes, perfect. Okay, thumbs up, excellent. Okay, so again, thank you everybody for joining. What I was hoping to do is, by way of introducing a lot of our work, is talk about clinical trials in general, just to give a quick overview of what are clinical trials, how do they advance patient care? How do we get from an idea that somebody might have to using that at the bedside, using that for actual patient care? What is that process like, and how do we shepherd things throughout that process? So first, I want to just underscore the need. So I'm a neurosurgeon. I deal with surgical diseases of the brain, and summarizing the need is complex, of course, but I think you can summarize it in 3 key words. So brain diseases are serious, they're common, and they're very, very expensive. So some of the major causes of human disability and death are in fact brain conditions. They're very common. I guarantee that either those listening, either themselves or know somebody in their family or their loved ones who is suffering from some of these diseases. So not only are they common, but they're expected to increase dramatically in the next 2 decades. From a public health perspective, they're also extremely resource intensive. So Alzheimer's disease alone is expected to cost the global economy more than $1 trillion by the year 2030. So there's an urgent need then to develop new, safe, and effective treatments. But what are the challenges? Well, as you can expect, there are many challenges. This is not an exhaustive list, but some of them include safety. So whatever treatment you want to develop has to be a safe treatment. Nobody will want to take or participate in any kind of treatment that is too risky, that causes harm. The second is this idea that in the brain sciences specifically, we've had tremendous advancements in the last several decades in our ability to make a diagnosis and to recognize something like maybe Parkinson's or Alzheimer's or a brain tumor on an MRI scan. But so far, we have not made that leap to developing new treatments and therapies and really impacting people's longevity with these diseases. The other challenge is time, but a lot of these trials and a lot of science itself takes a lot of time. There's regulation oversight, there's a lot of administration that happens. Now, one may have an idea, but then there are significant hurdles to getting that idea into the lab and ultimately into hospitals and clinics. And associated with that is costs. There are actual costs and there are opportunity costs and time as well. The clinical trials that were discussed, they take time and they go through a process, but nevertheless, they can be very time consuming. For us as clinicians, as scientists, it can sometimes be frustrating that we want things to move at a faster pace. But of course, there are checks and balances and very specific steps that science has to go through in order to make those treatments possible. So when we talk about clinical trials, and this is a slide borrowed from the FDA, which oversees clinical trials in the United States, but it's very similar here in Canada. When you talk about clinical trials, we talk about phases, and I'm sure many of you have heard of different phases of clinical trials. So it all starts with something called preclinical work. So work that's done in a lab, oftentimes in animal studies. And as soon as something looks promising, the first phase is going to be a phase 1 study. And the purpose of a phase 1 trial is really safety. It's to show for the first time in a human patient, whether it's a drug or whether it's a device, that something is safe. But that's not enough. Not only can something be safe, there has to be some signal, some promising sign that that drug or device may actually be doing what it's supposed to be doing. If that's present, if something is safe and looks to be effective, then a phase 2 trial is designed. And then really the purpose of a phase 2 trial is to try it in more patients, to see, was it just a fluke that in a small number of patients this looked safe, or that it looked effective? We need to try it in a larger number of patients just to show that, in fact, not only is this safe in a big number of people, but in fact, that it's effective. If that's successful, we move on to a phase 3 trial. So these are not only much larger trials, often done in hundreds or in thousands of patients, but the other thing that we do in late phase trials, such as a phase 3 trial, is we compare the treatment that we're investigating, that we're interested in, to what we call the gold standard. We compare it, for example, to the best and most ideal way that disease is treated, so that we can make a head-to-head comparison and say, does this new treatment or device, how does that stack up compared to the standard way that we treat a given disease? If it shows promise, and there are several checkpoints and specific methods that we use to establish that, then it progresses to a regulatory review at the federal government level. And then in our case, Health Canada has to take that information, weigh the evidence and say, yes, this is an effective treatment. It looks safe. It looks like it stands up well compared to the standard treatments. We can now offer it to patients on a standard basis. That is the process of clinical trials. So you have standard of care, just to summarize, you have standard of care treatment. You can take anything you want in that realm. You can take a treatment for depression, for example, or for Parkinson's disease. You can have medical treatments, non-medical treatments. In the case of depression or anxiety, psychotherapeutic treatments. You can think of this as the standard way to treat a patient. And then one has an idea. The first step, test it in the lab. See if you can work out the nuances of things like dose and the technical aspects of the treatment. If it looks promising there, you've met the threshold to now try it in human patients in a phase 1 trial. If that looks promising, you try larger trials in human patients, and ultimately you compare it to number 1, to how you treat patients in a standard fashion. If that passes muster, you advocate for coverage. You ultimately go through the process to have hospitals reimbursed for that, and you get support from hospitals and clinics to actually engage with it. And ultimately, that becomes standard of care, and you start the process all over again with something new that's promising and innovative, and that's how progress happens. That's how we constantly build new and more effective treatments over time, really over decades. So what are some key facts about clinical trials? As you can see, they're really the engine of developing new treatments. It's how we develop new drugs. It's how we test new devices. It's really the way that we make significant advances, not only in our understanding of diseases, but in their treatment as well. The common trend, the common theme for all trials is really that the first priority is always on patient safety. That's a question we ask ourselves at every single stage, whether it's phase 1, phase 2, or phase 3. Is the risk benefit there? Is the risk to the patient balanced with the benefit to the patient? So the patient priority is always there. All clinical trials, and Dr. Murray will speak to this in more detail, all happen in important ethical and regulatory frameworks. So there's a lot of oversight that goes into this. So one can't just do a clinical trial without having Research Ethics Board approval, in some instances without having Health Canada approval. So all of this happens in a very strictly organized framework that is run by hospitals, by universities, and ultimately by the government as well. Participation in all clinical trials is strictly voluntary. One can only participate after there's been a very detailed, systematic discussion with the investigators and with the clinical team. And on our side, as we do clinical trials, we really encourage a conversation between patients and their primary care physicians and their other physicians to ask, does it make sense for me to take part in this trial? Is this in my best interest? Is it really going to address some of the things that I'm most worried about and that I most care about? And finally, the risks and benefits must always be balanced in order to justify a trial. And I alluded this in one of my first slides. The question for sure is important. But arguably more important are the methods that we use to achieve those objectives and how we ultimately measure outcomes. So you can develop a treatment, a drug, or a device. If it's not safe and it looks like the risks are too high, it doesn't justify doing a clinical trial. Okay, so the risks must always be outweighed by the benefits. So a little word about us. This is Sunnybrook from way up high, and I underscore and I show this slide very regularly to illustrate a very important point. That even though I'm speaking here, and Mahil will speak, and Brian as well, Dr. Murray, we really speak on behalf of a tremendous team with whom we're incredibly privileged to work at Sunnybrook Research Institute. This is a huge institution. It's a huge hospital. There are thousands of people that work here, and within it is also the Sunnybrook Research Institute, where there are hundreds of scientists working, doing tremendous work. One of the great privileges that I have is working in a place where everybody's so laser focused on developing new treatments, on treating patients with the most challenging diseases, but also working on the next generation of treatments. And this kind of picture reminds me of that all the time. So one of the things that we're really interested in is the next generation of brain therapies, what we call emerging brain therapeutics. And there are different ways that we're looking at intervening in and influencing the brain in a whole host of different diseases. So one way is using sound energy and ultrasound, and I'll show you what that looks like. So focused ultrasound is a non-invasive way to use sound waves through the skull to directly target things in the brain that we know are driving very important and troublesome symptoms. Another way is electricity. We're using tools such as deep brain stimulation, which is a kind of pacemaker, but rather than the electrodes going to the heart, the pacemaker goes to the brain. So we jam the circuitry in the brain responsible for very important and challenging diseases like depression and anxiety. And then there's magnetism. And then we're using tools like transcranial magnetic stimulation to treat things like depression, PTSD, and OCD as well. All of these are different forms of energy that we use in a kind of precision strike way to influence circuits in the brain that we know are not functioning well enough in these diseases, to try to see, can we develop these therapies as a next generation therapeutics? So this is focused ultrasound, which I'll home in on for the rest of the slides. The focused ultrasound is very interesting. It's a helmet-like device that contains essentially 1,000 ultrasound transducers in a helmet-like array. And as I mentioned, we can use ultrasound to directly access parts of the brain that we know are malfunctioning or aren't functioning well. We can do this in a whole host of diseases. Why is this important? Well, up until 2012, really, we did not know whether this device that you're looking at right now would be safe, would be effective in any treatments, and we needed to test this in clinical trials. We knew we had the rationale for this. We knew we had the know-how from a technical and clinical perspective, but we needed to show that, in fact, in clinical trials, this was effective. So here we had a situation where we had a standard of care. The idea was tested already in the lab, and we needed to do some clinical trials in order to advocate and move this forward. And specifically, when you look at focused ultrasound, there's been a tremendous history of this going back for several decades. So this isn't necessarily a new idea, but this is something new in a clinical trial context. So we've known for several decades that you can use sound energy to generate lesions, precision strike holes in the brain for specific conditions such as tremor, but it hasn't really been tried in human patients. So what we wanted to do is replace existing tools that already exist out there so that we can do this procedure in a safer and more effective way. And one of the first diseases we wanted to treat is essential tremor. And this is what essential tremor looks like. So this is a common disease, a common condition that affects up to 2 to 3% of the population, predominantly over the age of 40. And the main way we treat essential tremor is with medical treatments. And medical treatments are effective, and they work very well in up to 2/3 to 3/4 of patients. But in about 1/4 to 1/3 of patients, the medical treatments are not effective, or they're not tolerated. And as a result of that, those patients for whom medical treatments don't work, they become eligible for surgery, for a surgical approach to specifically treat their tremor. So all neurosurgery for tremor, and again, I'm a surgeon, this is what I do, all surgical treatments for tremor are focused on the same region of the brain called the VIM nucleus of the thalamus. And you can do different things to this part of the brain. You can burn a hole in it with electricity. That's a procedure called radiofrequency thalamotomy. You can insert an electrode and jam the circuitry. That's called deep brain stimulation. Or you can use focused radiation therapy. That's called gamma knife radiosurgery. So all of those will have their respective advantages and disadvantages. DBS, for example, requires an operation. Gamma knife radiosurgery requires radiation. So they all have their merits and their relative pros and cons. What focused ultrasound allows us to do is a few key advantages. The first is in real time, we're able to monitor the temperature in the brain as we make the lesion. So every 3 seconds, we get feedback and knowing exactly how hot we're heating the tissue. The second part is we can visualize directly the procedure in real time. The whole procedure is done in an MRI scanner, and we can see the lesion develop here in the left side of the brain. So left and right are flipped on an MRI scan. You're looking at the left brain over here. And finally, we do the procedure with patients awake. So during the procedure, we test their tremor. Here on the left, you can see a patient trying to draw a spiral. And on the right, here they are after the procedure is completed while they're still in the scanner, writing upside down. They can see the spiral much improved. So this real-time feedback has really changed the way we treat tremor. To take a patient who looks like this immediately before their procedure, again, very disabling tremor, significant impacts on quality of life. Patients like this aren't able to look after themselves or dress, etc. In about a 2-hour procedure, we're able to generate a lesion on the brain so that patients look like this immediately after. Essentially, with a tremor on that dominant right arm, entirely gone. You can see still in the hospital gown. It just came off of the scanner and of the treatment with that tremor essentially cured within a couple of hours. So this has really been transformative in our ability to treat tremor. And this was the first time, what you're seeing here, the first time that we showed this in a human patient. This was a phase 1 trial. We had the idea in the lab. It's been known and theoretically thought of for several decades. So we thought to try it in a phase 1 trial in a human patient. After we got some experience, we designed the phase 2 trial and ultimately went on to a phase 3 trial together with 9 collaborative centers around the world. And we conducted a randomized control trial, which is a phase 3 trial, where what we did was just what I described earlier. We compared the standard of treatment for essential tremor with focused ultrasound, and indeed we showed that patient had a significant benefit with their tremor control. So just to give you a sense of how influential this has been in the field of essential tremor, we initially asked Sunnybrook for research ethics permission back in 2011 to do the first patient of our phase 1 trial. And we treated our first patient in 2012, about 6 months later. We published the results of our phase 1 trial, what we call a proof of concept study, in 2013. And then we spent several years doing the phase 2 and the phase 3 trials, ultimately concluding in 2015. We then used that data to appeal to the Ministry of Health and in the United States to the FDA to provide approval for the treatment, and ultimately secured that in 2016. So that period of time, that's 6 or 7 years from initial ethics submission to regulatory approval, is about 1/3 of the time it takes a typical device or drug to go that same trajectory. Okay, so that shows you just how quickly we were able to go through that process. So we did the phase 1 trial. We then did a phase 2 trial in a larger number of patients. And as you saw, we published the phase 3 trial together with our colleagues around the world in the New England Journal of Medicine. And that ultimately led to Health Canada approval. So what do you do next? You have Health Canada approval, you have this device, you see that it's safe, you see that it's effective. What do you do next? Well, ultimately, you want the Ministry of Health to support it, and you want hospitals to offer it as widely as possible. And that's what we did. What we did is we appealed to an organization known as the Ontario Health Technology Advisory Committee, and that's an organization known as OTAC. And what they do is they undergo an economic analysis, and they check, well, is this a cost-effective approach? Is this really the best way that we can treat tremor? Ultimately, they approved it, and they had this approved and vetted by another organization known as Health Quality Ontario, and ultimately made the recommendation to fund it at the ministry level. And we were privileged and happy to welcome Christine Elliott, who was at the time the Deputy Premier and Minister of Health. And this led directly to Sunnybrook being funded for treatment of essential tremor back in 2017. And currently, We treat approximately 50 to 60 patients per year with essential tremor as a result, again, of going along that progression from an idea phase 1 trial all the way to treatment. And again, last year, we were fortunate to mark the 10th anniversary of our first focused ultrasound case. We've now treated 350 patients, at least to date. We treat about 50 to 60 per year. It's fully ministry funded, fully reimbursed, and we're expanding this and have become really a provincial and national resource for patients to come from across the country to our institution to have this procedure done. And again, all started with an idea, with a phase 1 trial, going through that process. So I'll just briefly discuss, other indications that we're interested in. So we knew that making a lesion in the brain is very effective for patients with tremor. So it got us thinking, can we use the same procedure, the same technology to treat other diseases, other conditions where making a lesion in the brain may be effective? And we know that that's the case in patients with OCD and with depression, that there is a decades-long history of precision strike lesions in the brain of patients with those diseases. And now with focused ultrasound, we can replace the previous approaches and really do this in a much more safe, much more effective, much more precise way. And that's exactly what we've done since 2018 and 2019. We've been treating patients with treatment-refractory, treatment-resistant OCD. This is one of our patients who underwent this procedure. You can see on the MRI scans, these lesions on both sides of the brain. And here on the right, this is the result of the first few patients who have reached at least 6 to 12-month follow-up. You see significant reductions in their obsessive compulsive scores over a period of several months. From our perspective, this has really changed the game for patients with treatment-resistant anxiety. This is a day procedure, something that most patients go home either the same day or the next day. It's proven to be quite a safe procedure and now has gone on to more advanced clinical trials. So that idea going from, again, preclinical work to early phase clinical trials to larger clinical trials and ultimately to ministry approval, from our perspective, ultimately leads to changing the treatment of common disabling diseases. That the old model, for example, involved multiple days in the hospital, more invasive operations, maybe using toxic chemotherapies that aren't well tolerated, a trial and error approach in treating symptoms rather than treating diseases. Well, with something like focused ultrasound, we're converting this to same-day procedures, less invasive approaches, more tailored and personalized approaches that have the potential to really modify diseases in their natural history. So we're using that model now. And again, I mentioned earlier, very privileged to work alongside experts at Sunnybrook who are really advancing this along multiple different platforms, multiple different diseases. We now have clinical trials running across neurodegenerative conditions, movement disorders, cancer, including pediatric and adult brain cancer, and in mental health applications as well. For every single one of these, and I was just discussing essential tremor, for every single one of these, the hope and plan is to go through that phase 1, phase 2, phase 3 process, and to go again from an idea, from something that you want to see if it bears anything, all the way to standard of care treatment along the way. One of the things we're quite interested in also is cancer, and not only in new therapeutics, but in helping deliver chemotherapy to the brain. Just to show you the kind of process that we can go over time. This is work that showed that focused ultrasound, that very same tool that I was just describing, can be used to improve the delivery of chemotherapy to the brain. Initially shown in mouse models, shown here at Sunnybrook, and now progressed to human clinical trials, where just a couple of years ago, we published the first results of delivering chemotherapy known as Herceptin directly to the brain in patients with metastatic breast cancer. And this is one of those patients. I show you here on the left, brain tumor that is in an inoperable location of the brain that one cannot access this with an operation. With focused ultrasound, we're able to help enhance the delivery of chemotherapy directly to this region of the brain. And for the first time ever, show that we're actually able to engage the tumor with chemotherapy. So again, this was a phase 1 trial. And what you can see here, that we can actually visualize and see the chemotherapy in the brain. It lights up as yellow. And immediately before focused ultrasound, we did not see any chemotherapy within the tumor, pointed here in the arrow. And immediately after focused ultrasound, you see a significant lighting up of chemotherapy within the tumor. So this was, again, the first time that we were able to show that chemotherapy gets into the brain in a significant way in a phase 1 trial, so that ultrasound therapy can one day be used to enhance chemotherapy. So science is a process. It's slow at times, but at the same time, extremely promising, and something that really excites us and inspires us are the many questions left unanswered that require answering in order really to get to that stage where we can develop a therapy. But just to illustrate to you what some of those questions can be, specifically in the cancer space, we have to ask ourselves, well, which disease are we going to treat? At what stage of treatment? Where and at what stage of the standard of care should we apply something like focused ultrasound? What therapeutics should we be delivering to the brain? What should be the technical parameters? And finally, what outcomes are we measuring? What is meaningful to patients? What would a good outcome look like in a clinical trial? All of these question marks are the reason science exists. They're the reason research exists, so that we can really answer definitively each one of these question marks along the way. And that's why we've been able to sort of make a dent in a lot of these diseases. So over the last few years, our team at the Hartwell Center, and again, you'll hear from other members this evening, we've really managed to move the dial, or tried to move the dial on some of the most challenging diseases that we know impact our patients, whether it's using ultrasound in Parkinson's disease, in glioblastoma, which is a kind of brain cancer, in ALS, in OCD or depression, whether it's using ultrasound to deliver therapies or precision strike lesions in the brain. We're really trying to make a dent again in these diseases. There's no one out there who has not been touched by brain diseases, and there's no one out there who hasn't been affected and that will benefit from the kind of research that is happening at Sunnybrook all the time. So with that, I just want to thank the tremendous team with whom I'm very lucky to work with on a daily basis, including Mihaela, who you'll hear from next, Dr. Murray as well, who's our very close collaborator at the Research Ethics Board, and all of our collaborators, from our students to our techs. It takes not only a village, it takes a city, clinical trial work. But again, we're incredibly inspired by our patients to make an impact in their lives and the lives of their families. So happy to answer any questions.
Thank you so much, Nair. Such amazing work from you and your team. I'd like to now introduce our next speaker, Mihaela Lineth. She is the manager of Sunnybrook's Focused Ultrasound Center for Clinical Research. She has several years' experience in study coordination and regulatory medical affairs for phase 1 to 4 drug and medical device trials. Mihaela joined Sunnybrook in 2014 and coordinated the first pivotal trial in Canada for MRI-guided focused ultrasound for essential tremors. Under the Hartwell Center for Neuromodulation, she manages focused ultrasound blood-brain barrier opening trials for oncological brain diseases in adults and pediatric populations. She also oversees the day-to-day operations of the center. She has a strong commitment to advancing medical research, especially in challenging therapeutic areas. Clearly, she has extensive knowledge in the patient experience. We're so glad that she is here this evening to address some of the common questions participants in clinical trials might have. Without further ado, I'll pass the microphone over to you. Thank you, and give her a warm welcome.
Thank you very much, Lisa. I appreciate that wonderful introduction. I am going to start sharing my slides. Please let me know if you can see this. Everybody can see this? Okay, great. Sorry. So I'm going to be going through common questions that we get from potential clinical trial participants and how we answer them. When we are approached by a potential patient, or when we approach potential patients for participation in a clinical trial, we often find that they may have some restricted...
Apologies, Mihaela, I'm sorry to interrupt. Your slides are not up, so
No. Okay. Okay,
yeah,
one second.
sorry for interrupting.
Oh, no, that's okay. Let me just share a screen here. Sorry. Just give me a second here. Sorry for the technical difficulty. Let me just. My apologies. Trying to open it up.
Those look great, Mahela.
Sorry, I'm trying. So she's not.
They're up.
I think that Nir is still sharing his slides and I can't share. Sorry.
We can,
Someone
we
is.
can,
No,
we
no,
can see,
they're-
you can see your slides, Mahela.
Yes, it
Oh,
was
you
they're
working.
can
up.
see them?
Yeah,
Yeah, they were
it was
just up.
working.
They were
Oh.
up, not anymore. Yeah,
Okay, sorry.
you can start sharing again.
You can see that?
No,
Yes?
but now, yeah, now, yes.
Okay, great. Thank you. Okay. Yes, so as I was saying, patients are often approaching us about clinical trials, or we're approaching them about potential participation. We find that they do have some restricted knowledge about what exactly a clinical trial is all about, what the process entails, and what is their level of commitment that is required for them to participate in a clinical trial. So we want to answer the questions in a way that allows the patients to feel at ease and allows them to make the right decision for them, whether this particular trial is something that would work with what they're looking for. So there are various forms of research studies, and I'm going to be focusing on interventional trials. These are studies that involve human volunteers, and we're seeking to answer questions about a medical, surgical, or behavioral intervention. The researchers use scientific methods to determine if a new form of drug or medical device is safe and effective. Ultimately, clinical trials are designed to determine whether a treatment is more effective or has less harmful side effects than what's already out there. So in essence, a clinical trial gives us and the public evidence-based information. It allows us to advance scientific knowledge, develop new therapies and new technologies. It helps us understand the mechanisms of disease and how they can be treated. And it allows patients to have access to cutting-edge treatments, which they may not otherwise have access to. When we think of patients seeking clinical trials, we know that they're seeking often for therapeutic needs, but there is a great population of patients that are seeking to participate in the clinical trial for altruistic needs, as they want to help patients, other patients with similar conditions, and they want to also advance science. So the next natural question that we get from patients is, what is the purpose of this clinical trial that I'm being asked to participate in? So as researchers, it's important for us to highlight the exact purpose of the trial by providing, as always, honest, forthright information, using clear language, explaining difficult medical terminology, explaining the exact design of the study, and also what is the outcome that we're trying to measure. Managing patient expectations is also a vital part of what we do as researchers when we're speaking to patients. Some patients do come into a trial thinking that it might be a cure, and so we do need to kind of manage their expectations, and we want to let them know whether the trial is testing the drug or a medical device, what phase the study is in, as Dr. Lipman alluded to, the phases of clinical trial. Is the trial randomized or non-randomized? Is there a chance that the patient might be assigned to an arm that has a placebo medication and they may not get the treatment right away or delay treatment? So in clinical trials in Canada, for interventional trials, they are divided into phases, as we learned earlier. That each phase is answering a different question, has a different amount of patients that we need to answer those questions, and has a different level of scrutiny. For drug trials, there are, as Dr. Lipman showed, there is a phase 4 trial, which is post approval, but I will go through the first 3 phases for drug trials. In phase 1, we are looking at safety, and for drug trials, we're determining dosage, making sure that we are using the right dosage or the safe dose. It's usually conducted in 10 to 50 healthy volunteers and can take up to 2 years. Of course, the timelines for these phases can vary, but this is just a general idea. Phase 2 trials, we're looking still at safety, and we're starting to look for early indicators of efficacy. It could be done in under 100 patients or so with the medical condition that we're trying to investigate. And then once the study phase is completed, we can move on to the phase 3 trial, which now we're confirming efficacy, and we are testing the drug against the standard of care, the gold standard, or placebo, and this can be done in thousands of patients. Similarly, with the device trials, we go into several phases. The first being an early feasibility study or first in human. We're looking for preliminary safety here, and also device performance. Does it work as it's intended to do? This phase is usually involves about 10 to 30 patients. If this phase of the study is found to be safe, then we can move on to the second phase, which is a safety and feasibility. Here, we're looking also at early indicators of efficacy. We're also finalizing the device design, and it can involve up to 30 patients. It could be a little bit less or more than this. And then ultimately, we want to move into the pivotal phase where we're confirming the efficacy of the device, and now we're testing it against a control arm or a sham control or the standard of care. And the sham control is basically the device. It's the analogous to placebo. The next question that a patient will ask is, am I eligible to participate in this trial? Or can I participate in this trial? Who can participate in this trial? It is a very personal decision that patients make, whether they want to participate in a clinical trial. So we want to give them as much information so that they can decide whether they can participate in this particular trial. Depending on the trial, we're looking for healthy volunteers or patients with the disease in question. The protocol for the particular trial has a specific eligibility criteria that determines who or who cannot participate, and each study must have a particular set of criteria that have to be met for patients to participate. These are called inclusion-exclusion criteria. And when speaking to patients, it's important to let them know that these measures are not used to reject them personally. Instead, the measures are used so that investigators can test the intervention on a similar group of people. It ensures safety for all of the patients involved. It allows us to report on reliable results and allows the investigators to answer the question that is intended for that particular trial. So inclusion criteria are characteristics that qualify patients to be in the study. We look at things such as age, gender, or does the patient have the medical condition that is being studied? Whereas exclusion criteria are characteristics that disqualify or may disqualify a patient from participating. Age is a very important criteria. We want to make sure that we're not targeting a vulnerable population. For example, children or adolescents or an elderly population where the intervention may not be safe for them to undergo. Also, patients with pre-existing medical conditions, sometimes multiple conditions, comorbidities that may interact negatively too with the intervention. Patients that are on certain types of medications or multiple medications. Also, patients who have organ dysfunctions. These are kinds of the kinds of characteristics that may disqualify a patient from participating. It's important for us to inform patients that when they're being screened for a clinical study, they will undergo a series of assessments by way of imaging, blood work, questionnaires that determine their eligibility and whether they can proceed into the study. Oftentimes, the assessments and the tests continue throughout the study, and at any point, results from these tests may disqualify them from continuing the study. One of the next important questions that we often get asked is, what are the risks and benefits, in particular to the trial that I'm participating in? So giving patients a clear understanding, honest information about the risk and benefits, it's critical for them to make an informed decision. Even in drug trials that are providing a great alternative for hard to treat conditions or intervention, or for conditions that don't have a lot of therapeutic options, it's also important, or still important, to let the patients know that the intervention is still under investigation. And this is important for phase 1 to 2 trials, where we're not at the efficacy phase yet, and we're still looking for safety measures. In phase 3 or pivotal phase trials, we can provide patients trending data about efficacy and provide them available information about risk and benefits. Some risk and benefits that are general, there's a whole bunch, but some of the important benefits of participating in a clinical trial are, as I mentioned, patients having access to new treatments that they may not otherwise have access to. It allows patients to have focused healthcare. This is by way of extra imaging, direct patient to physician contact. It allows them to learn more about their health condition. It allows them to help other people with similar diseases and advance medical knowledge. At the same time, there are risks inherent with interventional trials that are under investigation. Unknown interactions of the drug or the medical device or the intervention with the body, the human body, or with diseases that the other comorbidities or medical conditions that the patient may have. Emerging side effects that can occur as we move forward into the phases of clinical trials, as we add more patients in the phases, new side effects do emerge. Unknown risk that we may not be aware of yet. Increased visits to the hospital that are above standard of care, burden on the patient, traveling time, those kinds of things. And ultimately that the intervention may not work. So once the patient knows about the risks and the benefits, the next question that we get asked is, what happens if I experience a side effect? It is important for us as researchers to let the patients know all about the side effects that are known to that point, and always always request from the patients or participants to alert the team about side effects, even if they feel it's not related to the intervention. This allows us to investigate side effects and monitor the patient and treat them immediately until resolution. It is also important for patients to recognize that if they do experience a side effect, depending on the level of the side effect, if it's getting a bit more serious, that the intervention may be paused. It could be, the drug dose could be reduced, or they may ultimately be advised to discontinue the trial if it's not in their best interest. We want to assure patients that clinical trials are designed with safety as a top priority. However, they do at any point also have the option to withdraw at any time, and it will not be held against them. As researchers, we really want to show empathy and understanding of the patient's concern and fears. We want to provide them with as much support and information as possible. Does the trial stop when a patient is experiencing a side effect? And basically, all clinical trials have safety and follow-up visits and assessments that are built into it so that we can reduce the impact of side effects or even the occurrence of side effects. There's also periodic reviews that we do on efficacy rates and side effects. This is done by data safety monitoring boards, as we mentioned earlier, ethics committee, regulatory oversight by Health Canada or FDA in the US and Health Canada here. Also, interim analysis to make sure that we are monitoring side effects and occurrences of these within each trial. The protocols often also have study stopping rules and decision points to make if we reach those criteria. These are built these are inbuilt measurements to safeguard participants. Ultimately, if the risk outweigh the benefits, the study can be halted and or modified. What happens after a trial? Patients often want to know where their next step is after they've participated in a clinical trial, and can they have access to the information? As we noted earlier, we do go through several phases. So, a patient can be participating in a safety trial, and the results of that study will dictate whether we can move on to a phase 2 or pivotal phase. Results can often be published in peer-reviewed articles, so patients may have access to the results of the trial through that. Participants may also be able to directly access the results of their participation, and this is very dependent on the clinical trial and the center. Also, the drug or medical device can be approved. As Dr. Lipsman showed, we have gone from early feasibility to approval phase, and those do take a while. So that's something that we also can remind the patients that, even though the drug is approved, it can take a little while for its use at a hospital level. And one important thing to discuss with patients is their continuing treatment after they participated in a clinical trial. Sometimes the drug or the intervention may not be available after the trial. In some cases it is, so it's important to discuss what their next steps are in terms of their treatment. So in answering all of these questions and all of these, and giving patients all this information, not only does it increase awareness of clinical trials and what we do, but it also provides patients, empowers them to make great decisions about their own healthcare. It allows them to be at ease. It increases health literacy and thereby reducing vulnerability of patients. Thank you.
Thank you so much, Mahalet. What an interesting wealth of information to share with our participants this evening. So before we hear from our next speaker, just a reminder to everyone watching to please send us your questions online if you have one, and we'll take the questions during our Q&A period. So now I have the pleasure of introducing our final speaker, Dr. Brian Murray. He is a professor of neurology and sleep medicine at the University of Toronto and Sunnybrook. He is also the chair of Sunnybrook's Research Ethics Board and head of the Division of Neurology. Our event this evening would certainly not be complete without Dr. Murray, and we are so grateful he is here to discuss how ethics help to guide clinical trials. Dr. Murray, the floor is yours.
Thanks very much for having me here today, and it's great to be with my my good colleagues. I'm going to speak today about how ethics can help guide clinical trials, and at the end of this short presentation, you'll be able to describe the structure and function of the Research Ethics Board, describe some basic ethical principles of research, describe the role of research ethics in protecting the participant for research. So Sunnybrook's Research Ethics Board, REB, our policy ensures that all research activities involving living human participants, human biological material, as well as human embryos, fetuses, fetal tissue, reproductive material, and stem cells derived from living and deceased individuals being conducted within or on behalf of Sunnybrook meet the current scientific, regulatory, and ethical standards for the protection of human research participants. Sunnybrook mandates the REB to independently review, approve, reject, propose modifications to, put on hold, or terminate any proposed or ongoing research activity that meets the criteria above. And there are many sets of eyes that look at any individual trial. We follow the policies and regulations of all these bodies. So the Tri-Council policy statement is a set of guidelines that are respected amongst the major granting agencies in Canada for the ethical conduct of research. We also follow International Council for Harmonization guidelines of good clinical practice in terms of running trials. Division 5 of the Food and Drug Regulations of Health Canada relate to drugs. Division 3 relate to radioactive tracers used in imaging. Part 4 of the Natural Health Products Regulations cover natural and alternative products. Part 3 of the Medical Devices Regulation regard devices. We also ensure that personal information is kept private under the Personal Health Information Protection Act. And we also follow U.S. Code of Federal Regulations so that we can participate in international trials with our American colleagues. And we have a set of standard operating procedures that Canadian Research Ethics Boards follows. So many guidelines to ensure that things are done right. The basic ethical principles that we follow boil down to 3 major considerations. One is respect for persons. So that means people have the right to be in the trial if they want, and they have the right to have their information used or not. Concern for welfare, and that is with respect to is the study safe, and is this a good thing for participants to be involved in? And justice. Everybody should have access and ability to participate, and we should be looking after all people. The Sunnybrook REB reports through the Medical Advisory Committee to the Board of Directors. So if something is unsafe, the information is conveyed to the highest level of the organization, and the safety of participants is paramount above all other considerations. We have other ethics boards at Sunnybrook besides the Sunnybrook Research Ethics Board. So we participate in a Ontario-wide process called Clinical Trials Ontario, for example. Our philosophy is that we are here not to protect participants from research, but we're here to protect participants for research. This activity, as outlined, is important. It's a serious business. People's lives are severely affected. It is costly both financially and personally. And Sunnybrook is helping invent the future of healthcare in terms of finding solutions to these problems. We want to help get these treatments to our participants. The Sunnybrook Research Ethics Board is required to consist of a number of components. There's a chair and a vice chair, and at least 10 members. We have approximately 25 to 30 on our board to manage the volume of activity that we have. We have to have members that have scientific expertise in the relevant research disciplines, fields, and methodologies. We have members that are knowledgeable in ethics, law, privacy, complementary or alternative healthcare where appropriate. We have to have at least one community member who has no affiliation with the sponsor, institution, investigator, and who is not part of the immediate family of a person who is affiliated. We have one member whose primary interest is in non-scientific areas. We have men and women, and a diversity of membership with consideration of race, gender, and cultural backgrounds. We meet monthly and have a meeting together collectively, but we also have activities that are delegated that we respond into the collective group. These volunteers provide about 12 hours of service per month, so it's quite a valuable contribution. These members help facilitate an amazing amount of work that's occurring at Sunnybrook. At Sunnybrook, at any given time, there are over 2,000 active research studies. Every year, we get approximately 500 new applications for REB review. About a fifth, 20%, require full board review, and that's where there's more than minimal risk. Many studies have delegated review, simple investigations like chart reviews or questionnaire studies. We manage over 1,000 amendments to the existing studies per year. We get safety reports. We consult with the investigators to help design safe protocols. We deal with privacy and participant concerns, and we ensure that we're accredited to the highest level. So this is just a graph here showing the activity that has exploded out of the Sunnybrook Research Institute, providing incredible advances. At our board meeting, we have a review of the general activities of the office, delegated issues, and we discuss new trends that we're seeing across different studies and have implications for us broadly. We look at about 8 to 10 studies in great detail at the collective board meeting. We get each board member would get an approximately phone book sized stack of material. And we went digital by 2020 to save trees. And this was an example of the amount of paper that we sent out for a meeting that's now digital. Our monthly meeting is about 3.5 hours. The primary reviewer of a study gets an additional phone book of more detail about the study, including a greater description of the drugs, and completes an itemized checklist to make sure all the T's have been crossed and all the I's have dotted. We review the study collectively. We provide an overview of the study and discuss items of major concern, and we come to consensus decisions about the right thing to do. We have input at every protocol, from medical, ethics, privacy, legal, and community around these concerns. And we try to be consistent and have developed a decision bank so that the same kinds of approaches are applied to everyone fairly. So every week we have something new in the Research Ethics Board that we hadn't considered before. And so this is an eye-opener in terms of the kinds of activities that are going on. So I just thought I'd provide a couple quick examples just to highlight some of the different kinds of things that we're thinking about. So one is around open data. So there's a move amongst medical journals to ensure that data collected in a research study is shared with other researchers to scrutinize and to also look for other signals that might not have been obvious. We do this at the behest of privacy. So we have to ensure that the best efforts are available to protect this information while sharing data. Digital apps on our phones are collecting data. We have to consider how that information flows and to whom it's flowing. Some devices are using geofencing, so they're able to track location of an individual completing a questionnaire. So we've had to consider how to manage that safely. Some studies have used services to track patient outcomes, and so we've been scrutinizing how this is done in a safe and ethical way. Artificial intelligence is changing our world. We are thinking about how studies are affected by these technologies. We want to ensure, for example, that populations are not overtrained to a specific study population, and making sure that the data is applicable to a broad range of persons. We're very careful about ensuring financial and other conflicts of interest are managed with each study. We consider who is to benefit and how can things be done safely and in the patient's best interest. We've had recent examples of imaging technology that can identify people by reconstructing facial tissues. So we've had to consider that in our reviews. And we've even had examples of oversight of research in warring countries. So every week we have some new issues that are very deliberately thought about to help ensure that the risk-benefit ratio for the patient is in the favorable state. So I just wanted to thank the group of people that I have the privilege of working with in this endeavor. Keitha McMorey, the Director of Research Integrity. Lisa, who's hosting tonight, is the Vice Chair. The Research Ethics Office, Chris Zaire, Catherine Perry, Naomi Chinda, and Dylan Kin, who are helping facilitate this work. The REB members, who are volunteers and contribute greatly to advancing these activities. And of course, the Sunnybrook research participants who make this all possible. And finally, I want to thank you for coming today to learn a bit about what we do and its importance. So thank you very much.
Thank you, Brian. And it's such a privilege to work with the Research Ethics Board under your leadership. And thank you for that great talk. So that concludes our formal presentations, but now we will begin the question and answer portion of the evening. If you'd like to submit a question, you can do so as we start with some of the questions that are posted already, and I want to thank the participants who have submitted them. So we'll start with the first one, and we're going in no particular order, but looking at how they're submitted in sequence. So what is the biggest challenge finding participants for clinical trials? And I was going to possibly direct this to Mihalis, but I will invite both Dr. Lipson and Dr. Murray as researchers to also offer their perspective.
Hi, thank you. What the biggest challenges that we see is getting the information out there that the clinical trial does exist. So having clinical trial awareness within the community is something that we're trying to increase. Other than that, trials have certain, as I mentioned, inclusion/exclusion criteria, and some of those are very specific, generally because we want to make sure that the trial is safe. So we find some challenges in getting patients that meet the entire set of inclusion criteria, although it's not impossible.
Yeah, I'll just echo that as well. It's definitely getting the word out that the trials are happening. Many of our trials are, for example, in an area like cancer or in mental health. These are areas where there's tons of interest and lots of trials happening. So patients, together with their caregivers and with their clinicians who are looking after them, have to sort of sift through all these options and make sure that they find the one that's right for them. So it's getting the word out, and it's matching, as Mihaela said, the criteria that the trial has with the particular patient that's interested in it.
Great. Dr. Murray, do you have anything to add from your own experience?
Nothing further. No, I think that encapsulates it pretty well.
Great, thank you. And that actually leads us nicely into the next question that was posted. How can I learn about clinical trials going on that I might want to join? So any tips or sources that possible participants can look at?
Yeah, so I think the first stop should be with your physician, so your primary care physician or your specialist. Speak to them, ask them, is a clinical trial right for me in my particular condition or the condition of my loved one? So that's the first stop, and that's a conversation they can have. There are also websites that are very helpful. One of them is run by the National Institute of Health, by the NIH in the US, and the website is www.clinicaltrials.gov.gov. And that's basically a repository of virtually all clinical trials happening throughout North America. So if you're running a clinical trial, you have to, or you should, or you need to register your trial. at that website with that website. So it's sort of a one-stop shop repository and you can filter there by location, by country, by city, and even by hospital. So you can put in Sunnybrook Hospital and you'll see all the trials that are happening here. And I also encourage you to check out the Sunnybrook website. There are some disease-related organizations. For example, the Alzheimer's Society of Ontario and of Canada has a list of ongoing trials in Alzheimer's disease. So all of those are potential sources of information.
Great, thank you so much. Now, this is, so we're taking a bit of shift in questions, but the question is, who pays for the trials? So directing to Meredith and Dr. Lipsman, who I'd like to respond to that.
Yes, our trials are often funded. If they're investigator-driven trials, we have grants and funding that we get from granting committees. And we also have trials that are funded by the sponsor, the makers of the device or the drug in question. Patients are not ever required to pay for a clinical trial. In fact, we can also give them some fees towards travel and their time, but all of our trials are funded.
Thank you so much. So Mahesh, I think this one.
Sorry, maybe I can just wait.
Yeah, go ahead.
Yeah. So absolutely, it is. So you have different buckets of funding that you can come from. My Health identified a very important, which is peer-reviewed granting agencies. Some of you may have heard of CIHR, NIH in the United States, et cetera. These are large government bodies that go to fund research. But an important bucket out there that is driving a lot of trial is philanthropy, and this is donations to the Sunnybrook Foundation and other foundations. A lot of the time, some trials are very difficult to get funded, especially the early phase trials, the first time that you're trialing something in a human patient, et cetera. Those are often difficult to get funded because there's not a lot of data available in human patients. Oftentimes, government agencies, they want to see some data first, whether it's promising, before they go in and invest in a larger trial. So that's where philanthropy really comes in, and sort of a discussion with the foundation and others. And we found that a lot of our most exciting early phase work has been driven by donors and by philanthropy.
Thank you so much, Dr. Lipsman. And Dr. Murray, I'm just going to go to you just if you wanted to add anything else, again, from your own experience.
No, I want to echo the importance of philanthropy in helping advance the work that we're doing at Sunnybrook. Donors have helped advance some amazing stuff, and there's plenty more that we can do. So it is a crucial element.
Great, thank you so much. Now, I think this question actually can go to any one of the panelists, again, based on your own experience with participants. How do trial participants feel about the experience after being part of a trial?
Maybe I'll start with that one. I think it's quite a testimony to the careful execution of these trials that people are generally satisfied. They are, carefully informed, carefully selected for an appropriate study, and things are done cautiously, thoughtfully, and we seldom have people that aren't happy with their participation, and most find that there's a value in doing it. Even if they haven't directly benefited, they know that there is altruism, that you may not benefit yourself, but you may help the broader population. So I think it's in general, most research participants are find it quite a rewarding experience.
Thank you, Dr. Lipsman. Mohammed?
Oh, yeah, I can't agree more. Mahala spends a lot of time with patients, and she'll obviously, speak to this as well. But I think the best, arguably, the best part of my job is dealing with, the research project that we do and shepherding patients through that process. And I'm incredibly, and I know I speak on behalf of our research team, we're incredibly inspired by our patients who participate in these early phase trials. And a lot of the time in trials where, there's not a clear, straight line that you can draw to benefit, specific, especially if you're doing a first in human trial where we're looking at safety, patients are often driven by, yes, a desire to see if there's treatment will work, but also by, by a sheer altruism, as Dr. Murray alluded to. And, to a person, we're thrilled to work with them. And it is really a partnership between the research team and patients in a trial. And they seem to be quite happy with taking part.
Yes, I wanted to add that we are often dealing with vulnerable patients, so we make sure that we're offering them as much information as possible, and that we're give them supportive, anything that they need in terms of support, answering their questions, navigating them through the hospital, doing whatever we can to do to make it an easier process for them. We understand that they're taking time out of their schedules to come to the hospital for visits, so we make sure that we're there for them at every step of the way, and minimize burden wherever we can.
Great, thank you so much. Our next question again speaks to participating in a trial, but taking a different viewpoint. What are the biggest things holding people back from taking part in a trial? And again, based on your own experience during your own clinical research.
Maybe you want to field that one first.
Me?
Yeah.
Yeah. I think it's having initially when coming and learning about or questioning about a clinical trial, not having a full understanding of the clinical trial and what's involved. Oftentimes, our clinical trials are also superimposed on standard of care. So when we explain the trial and we go through the design and what kind of commitment they're going to have to do, it often puts patients at ease, knowing that it's not as scary as they think it is. Also offering them the information that is available in terms of, as I mentioned in my presentation, about risk and how we minimize those risks or any all the safety measures that we take. It puts patients definitely at ease. Having that conversation with them and having actually multiple conversations with them if they still need it is important, because then patients do begin to understand exactly the process and how it can benefit them.
Great, thank you. Dr. Lipsman?
Yeah, I agree that a big part of our job is to educate patients about what is a clinic, just like what we're doing now. I mean, what is a trial? What is the purpose of the trial? I think that there's a lot of unknowns for sure when we come to it. As Mahil identified, a lot of the time we're dealing with patients with conditions that are scary, brain cancer and neurodegenerative conditions. So on top of dealing with all of that, patients now also are wondering, is this trial right for me? So I think that, the more knowledge is power, the more information we can provide, the better. But we're also conscious of information overload. So preventing too much at one sitting. So we have multiple discussions about consent and multiple discussions about the nature of the trial, and we make ourselves available as often as we can. So in that way, it sort of becomes a dialogue rather than sort of a one-time conversation.
Thank you so much. And Dr. Murray, this one is directed to you. So if someone wants to get involved with the Research Ethics Board as a community member, how much time do they have to commit?
So we've estimated that most board members commit around 10 to 12 hours per month of their time. It's quite flexible other than our group meeting, which is Tuesday afternoons. But I think, in general, members have found this to be an exceptional experience, educational, and interesting. We certainly welcome board membership from a variety of backgrounds. So not only with scientific expertise, but legal, ethics, privacy. So people are welcome to reach out to our board and apply for membership. And we collect a bit of information about what kinds of things you might be able to offer, and in particular, ensuring we have diversity across our board is a focus. So we welcome membership certainly.
And Dr. Murray, is there a way that the community members can connect to find out more information about possibly putting their names forward?
Yeah. So if you go to the Sunnybrook main website and look for the Research Ethics Board, there is a link there to community membership. So it should be possible to find the right person.
Great, thank you so much. So our next question is, what is the success rate for these trials, and how do you measure success? So this is a great question, and I'm going to direct it to our entire panel to tackle.
I do think it's a terrific question, and how we define outcomes is really depending on the phase of the trial that you're dealing with. So for a phase 1 trial, you're really looking at safety. And there, you're looking at what we call adverse events or serious adverse events. And there are different definitions of these that, we can get into or not, but essentially, is this too risky? Is what you're doing too risky to the patient? That's really the main outcome of a phase 1 trial. A phase 2 trial, you're really looking at safety still, but you're looking at more broad implementation. Are there challenges when you try to apply this treatment, this drug, or this device to a bigger population that maybe you didn't identify with a smaller population, and it only became apparent when you expanded the number of people that you enrolled? Still safety and still efficacy. And by efficacy, I mean, is it effective? And that will mean different things depending on what you're looking at. So for example, in brain cancer, we're looking at, does the tumor stay the same? Does it progress over time? Does it get smaller? In Alzheimer's disease, you can define outcomes in terms of, does the disease remain stable? Do patients still decline, but maybe at a slower rate over time? You can look at quality of life measures. So the amount of time that patients are engaging in hobbies, for example, or return to work, et cetera. So it's very important that when you design a trial, that you define your outcomes in a very specific way. Not only that you can then use methods in order to answer the result properly, but so that you tailor the results to the patient population that you're treating. So one of the things that we're really interested in at the Hartwell Center is to redefine how we measure outcomes in many different diseases, recognizing that for example, in some diseases like depression, the score that one gets on a paper and pencil test doesn't always translate into how that person's quality of life may be. That it may look like on paper, they're actually quite better, but they don't feel any better. And vice versa. It looks like maybe they don't look very good on paper, but their quality of life is much better. So how we define outcomes is really a key question. This is a great question and one we're actively interested in investigating.
Thank you, Dr. Lifsman, and passing to either Mahaleth or Dr. Murray, if you want to add.
I think that we want to do research that improves the health of our populations and provides better quality of life, and so that's ultimately what we're aiming for. We also have specific questions that the studies are designed for, and so we answer those questions. And from the perspective of the Research Ethics Board, has this study been done safely in as safe a possible as manner, protecting the interests of the participant and their privacy? So these sorts of things are also important in the execution of the trial. So all of those things are considered.
Thank you. And Mahaleth, did you want to add anything further?
No, I think that was a great response from both Brian and Nir.
Perfect. Thank you so much. So our next question is asking, in the research community, do you know of efforts being made to encourage diversity among participants so that results will be applicable to diverse populations? And again, I'm going to direct this to all panelists because I think all of the panel members can provide their own unique perspective.
I would start with the recognition that one of the core pillars of one of the core ethical principles that we abide to is that of justice, and so we want to ensure that there's subject selection that represents the broad community. In the past, there have been problems with misrepresentation of study populations, so only studying drugs in men, for example. Now there is, a concerted effort with every trial to address, is there fair subject selection here, and are we reaching the broad population? That also trickles down to the composition of the board. So we wanted to have a diverse board with lots of opinions and a variety of experiences. So we start there, and it is certainly considered in every study and an important issue that we address in every single protocol we see.
Yeah, I completely agree. I mean, I think research needs to reflect the society in which it operates, and clinical trials are no exception. We see this from the local level at our research ethics board, at the provincial level, and at the federal level. When we're applying for grants, it's absolutely imperative that we describe and we go through the efforts that we are going to ensure adequate representation of diverse populations in our work. So it's an important question and something that permeates everything that we do.
Thank you so much. And again, Mihaela, sorry, I just wanted to ask if you wanted to offer anything further from your own experience.
No, I think it was greatly answered as well. Perfectly answered.
Right. So we have another question that was just posted. So thank you for such an active discussion tonight. If one chooses to participate in a clinical trial, is there a risk that one will be in the control group and therefore receive placebo treatments and or medicines? And who wants to tackle that one?
So I can start, and I'll just say that it all depends on the study, the way that the study is designed. And this is all the upfront conversation that one would have with the investigator team and the clinical team. So if one is taking part in a randomized, controlled, placebo-controlled trial, that means that by definition, some of the participants may not be receiving what's called the active treatment. They may either be receiving a placebo or some other treatment. And if it's a randomized trial, then neither they nor the study investigators will know that until such time when everything is what's called unmasked or unblinded later on down the road. For a phase 1 trial, everybody gets the treatment. Everybody gets the what's prescribed or what the device that is given, and that's called a sort of a prospective open label. I mean, the label is open. Everybody knows what everybody's getting. It's only if one enrolls in a placebo-controlled trial that one gets that. So one has the potential of being in that arm. So it's an important question because it speaks to the kinds of things that you should be asking the investigators once they approach you to participate in a trial. Is there a placebo arm? Will I receive the active treatment? Will there be a period of blinding or unblinding? So all of these are important questions to ask.
And I just want to add also that when we're having the conversation in these phases where there are a control arm, we do definitely give patients the chance and the probability of what the chance of them being placed in a placebo arm. So they do have that up front. Some studies are designed that while they're in the study, they may not receive the intervention, but as soon as they're completed, they can receive the intervention. So there's those options as well.
I would say for any individual trial, there is a consideration around, how appropriate it is to be in a placebo arm or not. A lot of studies are trying to answer a question, and it's not actually known whether it's better to be in an arm or not. We consider that very carefully when we're denying a standard of care that is otherwise offered. Then there's increased scrutiny because we want to make sure that patients are not losing out on an otherwise effective therapy. And so the study is designed in a way to minimize that potential problem. And so I think every study is thought about very carefully in that lens, and the justification for placebo has to be really clear. And so that's something that's considered in the ethical review of all trials.
And even in cases where patients are assigned to a placebo arm, there's still the direct communications and visit. And so there's still that extra support from the researchers. And so patients still have access to that. There's a value in that as well.
So just adding to that question, if there's a trial and it seems that possibly one arm is doing better than the other, what actually happens in that particular case with the participants and possibly the placebo arm?
So one of the safeguards that we build into a lot of studies is a regular review of the data. So some studies have a data safety monitoring board where the final results might not be available, but these trends are looked at such that if it's very clear that one arm is better than another, then the study might be amended or terminated because then the risk benefit of participating changes. And so careful review of the data as it comes in is part of thoughtful trial design.
Thank you so much. Dr. Litzman or Mihalis, did you want to add anything to that particular question?
No, just to again, echoing what Brian said, that many trials have data safety monitoring boards, but I can think of one recent example in my field in neurosurgery where investigators who are experts, for example, in clot retrieval. So if somebody has, for example, a stroke, one can undergo a procedure where one is able to feed a catheter through the groin, get into the brain, and retrieve the clot to help with an acute stroke that's happening right away, to reperfuse, to open up the circulation of the brain. There were trials that were done for that a few years ago, and the results in the treatment arm were so compelling and so powerful that they stopped the trials and immediately fast-tracked this to approval. So there are mechanisms in place to make sure that if something looks, and that's the best case scenario, because that means you really hit upon a landmark treatment. And so there are mechanisms in place for both directions to look if the risks are too high or if the benefits are so good that we need to change what we're doing.
Thank you so much. And again, I wanted to thank the participants for all the fantastic questions that they posted. So we're just, before we end formally, just one more question to all panelists. So I'd like to go around our panel, and that is, what do you like most about working in research, and any final thoughts that you want to leave the audience with? So maybe we'll start with Mihalis.
Thank you. I have the privilege of working at Sunnybrook and working with these amazing researchers. As I mentioned, I was involved in initially in the clinical trial for focused ultrasound and lesioning, and had the privilege of watching that start from piloting phase to actually approval. So there's a satisfaction in knowing that what we are doing is making a difference, is working, and we are seeing that. So there's satisfaction in that for me personally. meeting with patients, speaking with them, hearing their stories, being involved in any way, small way, even if it's a small way, to be involved in their health and their decisions to, to participate in clinical trials. That's rewarding for me.
Thank you so much, Dr. Lipsman.
Many things. I would say I'm, inspired by our patients, and again, in the partnership that we work with them on advancing what we do. The science we find fascinating, and the opportunity to advance, advance the field and really be on the cutting edge of a lot of these therapies. And the incredible partnership that we have both at the hospital level, at the research institute level, and especially with, as I mentioned earlier, with our donors and philanthropists, and a special thank you to the Harkwell family, especially, who have given generously to the center, and who have allowed us to really advance a lot of this work and really pioneer a lot of these treatments. So thanks to them and all of our donors across the board who have really let us push the boundaries forward.
Great, thank you. Dr. Murray.
Well, I'm lucky because I have the benefit of looking at it from two perspectives. One is from the Research Ethics Board perspective, and it is a privilege to see so many volunteers actively engaged to help thoughtfully execute protocols at Sunnybrook, and I'm very proud to be part of this board. So that's one aspect, and then the other aspect is to be a clinician and be able to see the kinds of research being done here that is transformational, and it is just remarkable to see what can be offered and what the future could hold. So I think it's it's a really an inspiring place. I think Sunnybrook has really got some magic in terms of advancing important work, in particular in my field in brain sciences. So you saw tonight the transformational work that Dr. Lipsman has done, and there's tons of interesting things going on, and very happy to be involved in that success.
Thank you so much, all three of you, for your final thoughts. So before we wrap formally this session for the evening, I'd like to take a moment just to remind our participants of a few points. Please take a moment to complete our electronic evaluation form, because it will help with future planning of talks and topics for you, for our Sunnybrook community. You can also add your name to our mailing list for future talks. I again would like to thank our speakers for an incredibly informative evening, and really their display of passion and commitment for research. And this is why Sunnybrook invents the future of healthcare. So really thank them and their teams. Thank you so much to Colin Lynch from our board for providing introductions. And a big thanks to all of you for joining us for this discussion. And please be sure to check out our upcoming topics, which will be shared on the Speakers Series website at sunnybrook.ca. Thank you once again, and have a wonderful rest of evening.
Bye-bye.